All right, hello. Today we're going to be talking about Objective 11.3, meiosis. Explain what happens during each phase of meiosis. I want you to think about a normal human body cell. So that could be like a skin cell or a liver cell or a brain cell. That cell has 46 chromosomes. Now think about or a sex cell. Would your sex cells have 46 chromosomes? What would happen if your sex cells had 46 chromosomes? Like if a sperm had 46 chromosomes and an egg had 46 chromosomes, and then they combined, how many chromosomes would you have? The answer is too many. So no, sex cells can only have half the number of chromosomes, so that when they combine, it creates offspring with the correct number or 46 chromosomes. So today we're going to talk about the process that cuts that chromosome number in half, also known as meiosis. A quick review of some words, starting with homologous chromosomes. Homologous means chromosomes are chromosomes that have the same genes, and we got one from our mom and one from our dad. So this right here is actually two chromosomes. One of them came from dad, and one of them came from mom, and these chromosomes would have the same genes. Like this could be the gene for eye color, eye color. This could be the gene for hair color, hair color. This could be the gene to make your stomach and to make your stomach. The alleles could be different. Like this could be blue eyes, brown hair, blonde hair, but the genes are in the same spot. Interphase, remember the S phase of interphase is when DNA replicates? That's how they end up looking like X's, how we're more used to seeing them. So this is still a homologous pair of chromosomes, but now it has 1, 2, 3, 4 chromatids. This was a homologous pair of chromosomes with just one chromatid each, or two total. And then how do we count our chromosomes? Your chromosomes can be haploid or diploid. So in the normal body cell, they're diploid. That means they have both sets of homologous chromosomes. So this would be like from mom and from dad. This is from mom, this is from dad. But our sex cells only contain half of them. Like this one might be from mom, and then this one's from dad, and then this one's from mom. Let's talk a little bit about chromosome numbers. So haploid means half, and diploid means the full set. Given that information, can you fill in the rest of this chart? Go ahead and pause the video if you need to and fill it in. And hopefully these were the answers that you got. So if you know the haploid number is 23, the diploid number is going to be double that. 23 times 2 is 46. Sperm and egg are sex cells, so they have to have the haploid number, whereas the offspring, a normal organism, hopefully has the diploid number if everything goes well. By the way, cats really do have 38 chromosomes, and potatoes have 48, which is more than humans. You can Google if you're interested in knowing how many chromosomes a specific organism has. Meiosis is very similar to mitosis. So let's take a minute, look at this diagram. Can you recall the phases of mitosis? Can you recall what happens during them? Prophase, the chromosomes condense. Metaphase, the chromosomes line up in the middle. Anaphase, the sister chromatids separate. And telophase, we have two new cells. Meiosis is going to go through those same four phases, but it happens twice. This is how we get our sex cells, our sperm and egg cells. And remember, the purpose here is to cut the number of chromosomes in half because our sex cells need to have half the number of chromosomes. Before meiosis starts, the cell goes through interphase, just like before mitosis. And then it's going to go through the four steps, prophase, metaphase, anaphase, telophase once. That's why we have like a Roman numeral 1 in front of all of these. And then meiosis 2, it goes through the same four again. Now I have the Roman numeral 2. And we can see that we're starting off with a diploid cell and ending up with 4 haploid cells, or 4 sex cells with half the amount of DNA. Okay, during prophase one, the DNA is going to contain chromosomes, just like it did during normal prophase from mitosis. And then here's the difference. In prophase one, the homologous chromosomes are actually going to pair up. So like if this red thing right here is the chromosome from mom and this yellow thing is the chromosome from dad, it's going to pair up and this is called a tetrad. And this could be like chromosome 1 from dad, 1 from mom. Over here, this would be like chromosome 2 from dad, 2 from mom. And we also have crossing over occurring. So if you can see how the yellow chromosome is kind of touching the red chromosome right here, or here's like a zoom out of it, when they touch, their like parts actually stick together. And so the end result is like a red chromosome, but now some yellow, and then a yellow chromosome, but now some red. So those chromosomes actually exchange genetic information. Now the chromosomes are different than ever before. This is another source of variation. During metaphase one, the chromosomes are going to line up in the middle, metaphase middle, but what do you notice differently about it? They line up as homologous pairs. So here's the center line of the cell. They're lining up as homologous pairs. And when you look at them, you can tell that crossing over has occurred. During anaphase one is away. So this time, the whole chromosome separates. Instead of the sister chromatids, if we draw our line near the center line there, we have the chromosomes moving apart. And then during telophase, we're going to have two new cells forming. And notice that the cells are haploid. We started with 4 chromosomes. Now this cell has 2 chromosomes. This cell has 2. 4 reduced to 2, 2 chromosomes. Now we're going to pick back up and go through meiosis round 2. I want to point out that each cell has 2 chromosomes, but still has 4 chromatids. 1, 2, 3, 4. And there was no interphase. So there was an interphase before the first round of meiosis, but there's not another interphase before the second round. Because if there was, that would completely defeat the purpose of us trying to cut the genetic information in half. So pretty much nothing happens during prophase 2 except the cells are kind of there. And notice there is 2 because we just made 2. During metaphase 2, the chromosomes are going to line up in the middle just like they did in mitosis. So right here they line up in a straight line. There's our second cell, they're lining up in a straight line. And then during anaphase 2, the sister chromatids separate. So we cut it down the middle and there they are separating apart like that. And then telophase 2 is when the new cells form, and cytokinesis is when the cytoplasm divides. And what do we have is 4 different cells. They're haploid cells because they have 1, 2 chromosomes. And most importantly, they're genetically different. Like because of crossing over that occurred, look at how we have those different color combinations. This is another reason why you can resemble your siblings, but you don't look exactly like them. Because you could have this sperm or that sperm. There's a summary of the whole process together. Sometimes mistakes occur in meiosis. A common mistake is called non-disjunction. That's when the chromosomes don't separate. And this would be an example of non-disjunction during meiosis too. So there we have it separating correctly, but right here, this one is supposed to separate, but it doesn't. What's the result of that? Here we have a gamete with too many chromosomes, and here we have a gamete that doesn't have enough. This leads to genetic disorders like Down syndrome. Spend some time comparing mitosis and meiosis to really understand the process. Here we have the chromosomes that are separate, and here they're lining up as homologous pairs. When they line up in the middle in mitosis, it's in a straight line, but in meiosis, again, they're lining up as homologous pairs. When they separate in mitosis, they get pulled into single chromatids. When they separate in meiosis, they're still whole chromosomes. Because they were lined up as pairs, they stay whole chromosomes. And then down here at telophase, we have 2 new cells with the same amount of DNA. We start with 4 chromosomes, we end with 4 chromosomes. Meiosis is going to go through another round of division, and that's going to cut our genetic information in half. So up here, starting with 4 chromosomes, but ending with 2 genetically different chromosomes in each cell. This is a great video. I recommend that you watch it. And I'll leave you with this. The poor little chromosome says, "No, you never said there was a second anaphase. She's my sister. Ah." Oh, and I know this can be confusing, so please reach out to me with your questions. I'm happy to meet with you. Thanks, guys.